All right. Thanks everybody for joining and welcome to our current iteration of the NetPlace seminar, the last one of the year actually. And today we have Haley Merritt. Haley is a postdoc in London at the Network Science Institute. Her works involve working on brain science from a humanistic perspective, drawn from biological theory and complex systems science methods with consequences for social science. And she's going to be presenting us today exactly about that. So please, Haley, take it away.
Yes. Thank you so much for having me here today. I'm really excited to tell you about my research. I also have the videos minimized, so if you have questions while I'm talking or anything, feel free to interrupt. Um, yeah, so I'm going to tell you about some of the work that I've been doing since I started my postdoc a couple of months ago. But given that the discussion that I want to lead is on multidisciplinarity, I guess I should use this title with a bit of an asterisk. Um, because this is really about a multidisciplinary approach to human sociality. So the data that I'm going to be telling you about today is from the neural and psychological sciences. I'm a network neuroscientist by training, at least part of my training. Um, so the data are really about brain networks, well-being, social support. But given that before my PhD, I was working in the neuroecology lab, a lot of the theory that I use to inform the questions of my interpretations is coming from evolutionary biology and behavioral ecology. And because I have training in the social sciences and humanities too, I think it's really fruitful to be in conversation with a lot of the contemporary approaches to the contemporary social approaches to philosophy of mind. Given that I'm talking to you all, of course, I have to make relevant complex systems and network science. And that's where I get a lot of the methods that I think are really well suited to answering these really difficult multidisciplinary questions. And of course, all of the questions and the results, the interpretations, all have implications for the social sciences and humanities. So this is really drawing from a lot of fields, but it's not just that any of these fields are one part of the project. They're all interacting with each other and they inform each other, which is part of what makes this multidisciplinary, which we can talk a little bit about in the discussion. But yeah, this work is pulling from everything. But the key idea that really drives a lot of my research is the importance of our social relationships. So better quality social relationships are associated with better physical health. When I was in my PhD, I actually tried to see if I could find a physical health measure that wasn't associated with social relationship quality, and even things like bone density and the speed of wound healing. You're better off if you have better social relationships. So this is a really pervasive influence, perhaps more than you might think. Our social relationships are also associated with better mental health. This is perhaps less surprising, but pretty much every measure of mental health, you're better off if you have better quality social relationships. And not just our health, but also our cognition is associated with the social world. So people perform better at a variety of cognitive tasks when they have social support. But it's not just that there's this linear relationship between social support, social relationships, and our health and cognition, but really our cognition and our behavior are oriented around our relationships. And so I think these three studies that I've highlighted here show this in really different and interesting ways. So the study by Eshanjali and colleagues shows that when we're learning about new people, our memory of them is organized based on their relationships and not necessarily any traits of the individuals. And similarly, some of this work by Jim Cohen and Marlene Gonzalez shows that it takes us less energy to perform tasks and to respond neurally to different threats that we experience. So our social relationships are really important. So it's perhaps no surprise that our brains are really sensitive to our social relationships. Importantly, though, we all have different kinds of relationships. We have different experiences in the social world, and these differences can have huge consequences. So, if we think about evolutionary biology, developmental biology, it becomes really clear that over the course of development, as we're growing from childhood to adolescence to adulthood, our brains change and adapt to suit the demands of our relationships, the particular dynamics that we experience in the social world, such that we observe that brains with different social experiences are distinguishable from one another. This is really interesting and has important consequences for a variety of questions in human neuroscience. In particular, it tells us that it suggests that the brain organization that's associated with better mental health and better cognitive performance looks different in different social environments. So another way of saying this is that there is no such thing as what the one healthy brain looks like. There are lots of ways that a healthy brain can look, and to understand this requires looking at context and considering social relationships. So another consequence from this variability that comes from this ecological biology thinking is that this variability in brain network organization informs the conclusions that we can draw about how the brain supports cognition and behavior. This variability constrains the generalizability of brain-based biomarkers of different diseases, especially in the context of mental health. And it also gives some really necessary nuance to how we understand the normative trajectories of neurodevelopment. There are lots of different ways that are normal for a brain to develop because there are lots of different kinds of contexts that influence that neurodevelopment. And so I think the tools from complex systems, in particular network science, are really, really useful for understanding this variability and linking it to context. And so even though I'm only going to be sharing a little bit of results, I thought it would be fruitful for me to quickly tell you a little bit about how in network neuroscience, how we actually define the brain networks that I'm studying. So people are brought into the lab when we record activity from all parts of the brain simultaneously. Here I'm sort of showing time series of activity from 3 regions as a toy example, but we have activity from the entire brain simultaneously. Then, between all pairs of brain regions, we compute the Pearson correlation of their time series of activity. So this gives us a correlation matrix. And in network neuroscience, we call this correlation matrix resting state functional connectivity because usually this data is collected when people are just resting in the scanner. You can also have task functional connectivity. But we can interpret this resting state functional connectivity as telling us which regions of the brain tend to work together more. If there's a higher correlation, we often assume that these regions of the brain work together more. So then to link this to the theory from biology, we understand this variability in brain organization as telling us about differences in which regions of the brain tend to work together more. And so thinking about brain networks, these functional connectivity matrices, as phenotypes, then lets us draw on some existing work from evolutionary and developmental biology and behavioral ecology, and it gives us suggestions for the kinds of questions that might be especially informative for understanding the link to health, well-being, cognition, behavior, and things like that. So this is really tying together lots of different fields to get at this question. So the particular research question that I'm going to be talking about today, and that has guided a lot of my work during my PhD and into my postdoc, is how are brain networks organized differently across social environments? In particular, today I'll be talking about different social environment dynamics because, of course, the social environment isn't static. Um, and also, a really important question when we're thinking about ecology is the consequences of these differences. What does it mean for that environment, for a person to have that particular phenotype? So to get at this question of brain network differences across different social environment dynamics, the first step is just to identify these differences, to identify distinct trajectories of social environment dynamics. And I'll get into the data that we're using to do this a little bit on the next slide. Once we have these distinct trajectories of social environment dynamics, we want to look at their differences in mental health outcomes. So this sort of gets at the idea of consequences. Um, and then also, because I have training in network neuroscience, it's interesting to me to then study the differences in brain network connectivity across these different trajectories. So the data that we're using to do this comes from the Adolescent Brain Cognitive Development Study, which all in all is collecting data from about 12,000 adolescents from across the entire US. I think there are 21 different data collection sites. The particular subset that we're using has just under 5,000 adolescents, and it has a variety of different measures. So there are social environment measures that are collected every single year. Um, we're using 10 in particular. There are 17 measures of mental health that are collected every other year. There are also 3 additional composite measures of mental health that I'll tell you a little bit about when I get into those results. And then we have resting state fMRI scans from the first time point. So this is a subset of this larger data consortia. All in all, they're collecting about 10 years of data, but this is from an early release, so we just have 4 years of measures and neuroimaging data from the first time point. So the idea is to then use these social environment measures to see people's trajectories, how they change, how their social environments change over time, and then cluster the trajectories so we can look at differences in change in mental health and in brain network organization. So to do this, we calculate the correlation between all people across all measures, across all time points to get a similarity matrix. I've strategically organized the similarity matrix to hint at the cluster structure that we get. Um, we get the clusters by applying the Louvain algorithm for modularity maximization on the similarity matrix. So then now that we have the clusters, we can look at their particular trajectories of social environment dynamics. There are three main clusters that come out from this analysis. Um, I'll say that we ran the community detection algorithm 1,000 times and then computed consensus communities. So these are pretty robust clusters. In general, we see that cluster one, this line in pink, experiences more conflict and less support overall over time. So this is like a lower quality social environment, less supportive. Cluster two in green experiences lower risk, higher support, also high religiosity. So this is like a higher quality social environment. And then cluster three in blue is sort of intermediate, high risk, but also high support, and their lower religiosity. We also looked at a couple of different measures that might be confounding this, so like socioeconomic status, and we found that cluster three, the sort of intermediate cluster, has a significantly higher socioeconomic status. They're also like less further along in their pubertal development. So recall these are adolescents. They're like 9 or 10 years old at the first time point. So not as many of them have started puberty, but there are no differences in age or gender across these different clusters. So now that we have these clusters of social environment dynamics, we can look at mental health, some of their outcomes. So to orient you to this figure a bit, I'm using these colored shapes to indicate which measures are used in the calculation of the composite measures, which are the three at the top in bold: total problems, externalizing problems, and internalizing problems. And scoring higher means your mental health is worse. So you exhibit more symptoms attributed to, for example, anxiety and depression. So what we see from this figure is cluster one, which you'll recall has the lower quality social environment, has worse mental health. This is not especially surprising. We know, as I said at the very beginning, that social relationships, the quality of the social environment is strongly linked to mental health. Um, Similarly, cluster two, which has the higher quality social environment, experiences really good mental health. They're doing pretty okay. Also, pretty stable mental health over adolescence, which is pretty impressive. Cluster three, again, is sort of intermediate. But then when we look at the slopes of the lines, they exhibit the biggest declines. So their mental health gets worse faster than the other clusters do. So we see these divergent mental health outcomes across these different trajectories of social environment dynamics. Um, and then in this last set of analyses that I'll present today, because this work is still in progress, we also see differences in brain network connectivity. So what I'm showing in these three matrices here is basically differences between each pair of clusters. So on the top left is cluster one minus cluster two, and then cluster one minus three, and then on the bottom is two minus three. And I'm only showing colors if there are statistically significant differences. Um, and I know that many of you are not neuroscientists, but you don't need to be a neuroscientist to see that we see differences distributed across the entire brain, um, which is really interesting. And I also have this figure here sort of showing, given all of these statistical comparisons, which cluster has the highest functional connectivity between regions. And I wanted to highlight this distinction that I have made, like, informed but semi-arbitrarily between somatosensory and higher order cognitive brain systems. So somatosensory systems are brain systems like auditory cortex, somatomotor cortex, visual cortex. These are the parts of the brain that historically and traditionally we think of as processing like low level sensory information about the body. You're processing sound, you're processing visual input. These are the systems I've sort of drawn the red boxes around, versus the higher order cognitive systems like singular opercular, central parietal, default mode network, frontoparietal, retrosplenotemporal. These are the brain systems that we often think of as doing the heavy lifting of cognition or intelligence. They're the ones that are making associations between different inputs. They're processing the information that they receive from other parts of the brain and the body. These are the ones that are more often associated with impressive cognitive capacities. But what these results are telling us is that there's a trade-off in the connectivity that involves both the somatosensory systems and also the higher order cognitive brain systems. And so that a more like traditional interpretation of the brain might be a little confused by this. But there's been an emergence in work in human neuroscience in the last couple of years that is leaning into this idea that brains aren't just for cognition. They do provide our cognitive capacities, but also our brains regulate different physiological like rhythms that we need to live, but also in order to successfully interact with people. Um, so this sort of helps us understand why we're seeing interesting trade-offs in connectivity between the what people have historically called like lower order somatosensory and higher order cognitive brain systems. So there's a much richer picture to be told that I'm only scratching the surface of here for the sake of time, um, because I wanted to sorry, because I wanted to sort of be able to weave together multidisciplinary perspectives. So, like I said, this project is still in progress. I'm hopefully going to present it, if it all goes well, at Complenet and NetSci and maybe FENS if any of you will be there. But if you can't wait that long, we're hoping to have a preprint out early next year. But there are still a couple of analyses still to do. So given the nature of the longitudinal data, we're interested in analyzing which aspects of social environment change contribute the most to changes in mental health. So thinking about this in terms of interventions, we could ask what kinds of features of the social environment, what kinds of changes in the social environment are most associated with an improvement in mental health, and then we can try to implement that for adolescents who are struggling with their mental health. But given this importance of context that I was stressing at the beginning of the talk, we're also planning on looking at how the brain network associations with mental health look different across the different cluster trajectories. Um, Again, like the importance of ecology and context. So some of the take-home messages from this like super fast overview of some ongoing work is one that brain networks are organized differently in different social environments. Um, this is a really important feature of a lot of the work that I do because it has really big consequences for how we then study the neural underpinnings of mental health and how we think about human social behavior and its neural underpinnings. And I always really stress the importance of social relationships. This is super clear in the literature, but I think also in our own lives, especially after the pandemic. So there's lots still to do with this project, but also with a variety of other things. So the neuroimaging data is just from one time point, but it's also really worth investigating diverging trajectories of brain network reconfiguration. So how does neurodevelopment look different across different social environments? In some of my own work, I've shown the importance of looking at live social interaction. So when we're collecting this neuroimaging data from people, they're in isolation in an MRI scanner, but being in live social interaction and studying the whole social system with all of the interacting social agents is really critical to understanding how actually the brain, like in real time, contributes to successful social behavior and the dynamics of social support across a social network as well to connect this to network science again. Um, but also this field is evolving really, really quickly, and the questions are becoming more and more complicated. And so developing really innovative methods to ask questions that are informed by a lot of different fields is really, really critical. And so this is why I like talking to complex systems audiences, because you are really good at coming up with really cool methods to ask really cool questions. Um, so, like I said, our social relationships really, really matter. We really, really need each other. Go give your friends a hug, take them out for a beer or something. That's all the results that I have for today because I wanted to give us plenty of time to chat and for questions. But of course, this work wouldn't have happened without my own academic social environment all over the world. And I included a QR code to my website if you want to find any of the PDFs for any of the papers that I noted in the bottom bit of the slides. So thank you. I'm happy to take questions about the science.
Wow. Yeah. Thanks indeed. Very interesting. Um, yeah. I'm very curious. We'll start with looking for questions from the audience if anybody has one to begin with.
Hmm. I'm willing to ask one, but I would like to thank like Haley first. Like it was a really fascinating like talk. Like my question would be like more or less about like we saw like different kind of like as like parts of like a social interactions, like how, for example, like putting some of them together, some of these attributes together, like connect with the like the interactions in brain. For example, I think like maybe like gender would be really important, but like the number of like the social interactions would be important. Like how these things like the intersection of them like would be really important in terms of like the connections in the brain.
Yeah, for sure. So I think also like there are some like tiny gender differences in brain network organization, but the effect sizes of those differences are usually dwarfed by a lot of these social measures. But I think it's also worth noting that the way, especially like adolescent boys and girls navigate the social world is really, really different. Um, and the way that they like form relationships, the way that they rely on their peers, especially given the fact that girls tend to mature faster and are hitting puberty before boys, really changes their experience with their peers. So it's not uncommon for researchers to observe differences in some of these social measures. Um, I'll go back to um, one of the slides. Oops. Where I show some of these social measures. So I would expect like maybe some differences in like some of the more peer-oriented measures, like maybe community risk, some of the school-related measures, or pro-sociality. But I wouldn't really expect such gender differences with like the family measures, like family conflict or parental monitoring. So something like gender is going to play a role in some ways. But as I said, when I was talking about these different trajectories, we don't actually observe gender differences across the clusters. So they're pretty evenly distributed in terms of what we see here.
But what if you like connect the independence and family conflict like together, meaning like you see like intersection of like these two like at the same time? Like, do you see like differences in those things?
So we haven't looked, I haven't looked at just like a subset of these measures before because I'm usually trying to get at like a very broad
Mhm.
uh like representation of the social environment. But there have been thousands of papers published with this data set, so I'm sure that somebody has done this. But
Mhm.
I don't know off the top of my head how exactly it pans out.
Okay, thank you so much.
Mhm.
Okay, we have time for maybe one or two more questions if anybody has one. If not, I have a question of my own, or maybe a remark. I'm curious about, in the end, when you were talking about sort of the future work, in regarding to the temporal aspect, or especially in relating to how it reacts to different sort of environmental changes. And this is two parts. One is the current data as it stands temporal, as in do you see these fluctuations over time and having the snapshots of the different networks? And also in regards to how would you go about studying maybe long-term changes based on environmental or external circumstances?
Yeah, so yeah, the data are temporal. We're using just a subset of this. So this whole data set is being collected over 10 years. It's still in progress. What I'm using here is from a really early release. So it's not all of the data that's been collected, even though it's most of the people. So eventually, when all of the data has been collected and pre-processed and is ready for analysis, there will be neuroimaging data every year for 10 years. I just don't have that right now. Um, but we're working with some collaborators to get access to a more recent release, which will have more neuroimaging data, which will let us get at this idea of changes in brain network organization over development in concert with the social environment. Um, there are so many ways that you could study this. What is by far the most common, but perhaps a little unsatisfying for us complexity scientists, is for people to treat the edge weights in brain networks independently and just perform as many statistical tests as you have unique edge weights. So you look across your group of people and ask, okay, is this edge weight different across groups? Okay, is this edge weight different across groups? Given that we're calculating the edge weights using Pearson correlations, and given that this is a spatially embedded network, we expect adjacent nodes to be somewhat similar to each other, so there's some spatial autocorrelation. So these edge weights are not independent from each other. And so this traditional, albeit very easy, very accessible approach to studying differences, um, this like traditional approach as a complexity scientist, I find really unsatisfying. So one way that you can leverage the fact that these are networks that are spatially embedded, that have spatial autocorrelation, that have non-independent edge weights, you could use multi-layer networks, where each layer is a time point and you can couple them based on your social environment measures. You could perform community detection. Um, there are lots of other options that you could do. I've tried a couple, some work better than others. Multi-layer networks, love them, super computationally expensive. Um, but I think this is also a place where there's a lot of really promising opportunities for collaboration between people who have expertise in complex systems and the psychologists and the neuroscientists who have these really interesting questions, but whose methods aren't always up to the task. Um, so all of that to say, like, I have some ideas because I'm like at the intersection of the space, but also like if you have ideas, like speak to some potential collaborators. It's they love complex systems in psychology. They're a big fan of our methods. Uh, yeah.
Yeah, it's quite interesting because even the ones in theoretical complex systems and network science, they wouldn't even be able to also give you a concrete answer of exactly how to analyze it, right? It depends a bit on the underlying assumptions, and you would need to sort of curtail it a bit, but
Sure.
definitely an interesting challenge for sure.